So this is where, excuse me, this is where we stopped last time. So we were talking about this hemoglobin saturation curve. So let's look at it some more. So if we look at the hemoglobin saturation curve, first let's look at the axes. So over here, look, this is just the percent of hemoglobin that's bound with oxygen. So right here, no hemoglobin has any oxygen on it. But look, here 20% of the hemoglobin has oxygen. Here 40% of the hemoglobin, 60%. So up here, 100% of the hemoglobin has oxygen bound to it. And so it's saturated, and that's why it's called a saturation curve. Well, remember, hemoglobin can bind 1, 2, 3, or 4 oxygens, up to 4. And it just depends on how much oxygen there is, how much gets bound. So when we have very little oxygen, very little is going to be bound. So look, if I have a partial pressure of oxygen that's 20, not much hemoglobin is going to have oxygen on it. Basically this much. But when I get here, this much has it. And when I get to 100, it's about what it is in the lungs. Remember, it's about 104, 105. 100% of the hemoglobin gets filled up with oxygen. Well, when we look at this curve, remember, it's not a straight line. It's not like this, which is what we probably would expect it to be. Because we would expect that for every oxygen we have here, we're going to add it to hemoglobin. So if we got 20, we should have 20. If we got 40, we should have 40. In other words, we expect it to be a straight line at a 45-degree angle. But it's not like that. Because hemoglobin has this property, and I didn't use this word last time, and you don't really have to know it. Oh, never mind. But hemoglobin changes shape. And what makes it change shape is oxygen binding. And so when oxygen binds, it changes the shape. So our curve does not look like that. It's this S-shaped curve. S-shaped, like that. It's called a sigmoid curve. Well, the only reason we talk about this is because it makes hemoglobin much, much more efficient at picking up oxygen. Look how steep this line is right here compared to this one. Remember, this is a 45-degree angle. This one might be an 85-degree angle. And so what happens is oxygen binds really, 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 really fast. So a lot of oxygen binds without a big change in how much oxygen there is. And it's really fortunate that it's like this. Because look, this curve makes it bind faster. So remember, the most oxygen we have in our lungs is about 104, 105. That's it. Well, because this curve is shaped the way it is, look, 100% of the hemoglobin binds to oxygen. Well, what if it were this other curve? How much oxygen would I have to get to 100%? So here's 100%. I would need this much oxygen. What is that? So there's 80, 100, that's 120. That's like 140. In other words, the partial pressure of oxygen in your lungs would have to be 140 to fill this up to 100%. That's just not possible. And if that's not possible, this wouldn't be possible. But because the curve is shaped the way it is, it fills up much, much, much faster. And so we can fill up 100% of our hemoglobin with oxygen at the same level that we have in the lungs. It also, though, means we can deliver oxygen faster. Look, we deliver it faster because it's steep. Does that answer your question, Letitia? So look, here's this curve. And we cannot change the shape of this curve. That's just the shape. That's the way it is. But what we can do is we can shift it. So let's look and see what happens if you shift this curve. Before we do that, though, I want to tell you sort of an analogy. So my analogy is, let's go to a blank. So if we look at this, I can make, everybody in here knows what a magnet is, right? So a magnet is attracted to metal. I can make a magnet. I can make an electromagnet. And they're really, really easy to make. I can take a nail, and all I have to do is wrap this nail in wire, and then I can run a current through the wire, and I'll make an electromagnet. And that nail will become a magnet. And if I turn the volume or the voltage down, it will be a weak magnet. So what does that mean, a weak magnet? It means it's not very attracted to metal. So if I took a bag full of metal shavings and I dumped them out here on the floor, right here where this magnet is, well, it's not a very strong magnet, but some of them are going to bind to the magnet. But not all of them, because it's not a very strong magnet. But look, if I take that same nail, same wire, same situation, but I run a bigger current through it, it becomes a strong magnet. And so all of those little filings, now this thing is a lot more attractive. They're going to jump up here and they're all going to attach to this thing. And there's going to be very, very, very few filings that are not attached. Does that make sense to everyone? So look, what if I take this exact same magnet and I turn the voltage down? What do you think is going to happen to these filings? Are they going to stay stuck up here when it's a weak magnet? No, they're going to fall off. Well, now I'm going to turn the voltage back up. What's going to happen to the filings? They're going to jump back up here again, right? So look, I can adjust how strong this magnet is. I can adjust how much the magnet likes the filings. And there's a word for that. And the word for liking is affinity. So if I look at a weak magnet has a low affinity. It does not pick them up very quickly, and it does not hold on very strong. It's a weak attraction. And so a weak attraction means it comes off really, really easy. It doesn't pick up very fast. But look, if I look at this strong magnet, it has a high affinity. In other words, it's going to grab those filings really quickly. It's a strong attraction, and they're going to be held on really tight. Everybody understand this analogy? Any questions about it? Well, hemoglobin is like this magnet. So remember, hemoglobin's got 4 peptide chains, and in the center of every one of these is a heme. And in the heme, that's where the oxygen binds. Remember, there's an iron. So sometimes hemoglobin acts like this. And what's going to happen is the oxygen is going to fall off. In other words, hemoglobin has a low affinity for oxygen. Doesn't really like it that much right now. But sometimes hemoglobin acts like this. And what it has now, it has a high affinity for oxygen. And if it has a high affinity for oxygen, oxygen is going to bind really quickly, and it's going to bind tightly. So oxygen is going to bind really, really, really fast and tight. Everybody still with me with the analogy? So remember, with my magnet, what I was changing was the voltage that was going through there. So something has to be changing for hemoglobin to sometimes act like a weak magnet and sometimes act like a strong magnet. Does that make sense? So let's look at our graph. So here is this curve. So remember, it looks sort of S-shaped. It likes, yeah, that's not a very good one, but anyway, let's redraw it. So here's our curve. Let's still use black, but anyway, it's sort of S-shaped. Much better. So look, I'm going to pick just a spot on here, and I'm going to say that the partial pressure of oxygen, sorry, wrong thing. The percent of hemoglobin that's bound to oxygen is 50. So 50%. And look, I don't know what this is, but this is how much oxygen that took to fill this up to 50%. It took that much oxygen. So the partial pressure of oxygen, I don't know what that number is, but let's say it's 40. Does it make sense? Looks just like our curve we had before. 50% of the hemoglobin bound to oxygen at this partial pressure. But let's look and see what happens if I move this curve. So let's move it to the left first. So our curve is going to have the same shape. And I want to fill it up to 50%. But look, it only takes this much oxygen. It only takes 30 to fill it up to 50%. So if I move it to the left, what happened? What happened to affinity? Did affinity go up or affinity go down? Does hemoglobin like oxygen and pick it up faster, or does it not like oxygen and pick it up slower? So it's going to be like a strong magnet now. So remember, with a strong magnet, we have a high affinity. So in other words, if the curve moves to the left, hemoglobin's affinity goes up. And if the affinity goes up, it makes it easier to pick up oxygen. It makes it easier to load oxygen. Does that make sense? It's like a strong magnet. Strong magnet, it was easier to grab the filings. Strong magnet for hemoglobin makes it easier to grab oxygen. Make sense? But let's look at what happens if I move it this way. I'm going to move it over here. So I'm moving the curve to the right. So look, I'm going to move it to the right. And now, look how much oxygen it's going to take to fill my hemoglobin up to 50%. It's going to take 50. In other words, it takes more oxygen just to get to the same amount. Does it sound like hemoglobin likes it as much? It doesn't. It's like the weak magnet. And remember, the weak magnet has a lower affinity. And so if I move the curve to the right, affinity goes down. Well, that doesn't make it easier to load. That makes it easier for the oxygen to fall off. It makes it easier to unload the oxygen. Make sense? So, I move to the left. What does that mean? I move to the left, what happens to affinity? Affinity goes up. If affinity goes up, does it make it easier to load or unload? If affinity goes up, does it make it easier to load or unload? It makes it easier to load. If I move the curve to the right, affinity goes down. What is that going to make it easier to do? Unload. So in other words, if I shift it back and forth, just like that magnet we had, sometimes it's easier to load oxygen, sometimes it's easier to unload oxygen. So now we have to talk about what makes it shift back and forth. So let's go back to our PowerPoint. It's going to take a second, I'm sorry, but I wish you could just go backwards, but that's not what this system does. So, look, here we are. Exactly what we just said. So, if we move the curve to the left, affinity goes up. And if affinity goes up, it's easier to load. If we move the curve to the right, affinity goes down. And if we move it to the right and affinity goes down, it makes it easier to unload. Is everybody understanding this? So, if the curve shifts to the left, hemoglobin is going to increase its affinity for oxygen. It likes it more. It's going to grab it faster, so it's easier to load. But if the curve shifts to the right, hemoglobin's affinity for oxygen goes down. It does not like it as much. And so it makes it easier for it to come off, easier to unload oxygen. So here's our normal curve. There's the left shift. Look what happens to affinity. So what is it easier to do if it shifts to the left? Load or unload? It's easier to load. But look, here's the other one. If it shifts to the right, affinity goes down. And if it shifts to the right, what does it make it easier to do? Load or unload? Unload. Everybody getting this? Sounds like you are. And again, here's the same thing. So a left shift increases affinity, easier to load. Right shift decreases affinity, easier to unload. So now all we have to do is figure out what is making it move. What makes that happen? So there are factors that affect curve shifting. These are the things that have caused the curve to shift. So temperature, just plain old temperature, how hot is it? That makes the curve shift. pH of the blood, which is the same thing as saying the hydrogen ions, that makes it shift. How much carbon dioxide makes it shift. And then there's one other thing which is called BPG, which is also going to make it shift. So let's look at each one of these individually. Let's look at temperature first. Here's what happens. If you increase the temperature, the curve shifts to the right. We already know what happens if the curve shifts to the right. What happens to affinity? Affinity goes down. We're going to decrease affinity. And if we decrease affinity, what does that make it easier to do? Load or unload? It's easier to what? Unload. That's exactly right. Easier to unload. Well, most of us think all of our body is the same temperature, but it's not. First of all, let's think of this. There are two places in the body. One place we want to load oxygen. Where do we load oxygen? What part of your body do we get oxygen at? We pick it up where? In the lungs. We pick up oxygen in the lungs. That's where air is. And then where do we unload oxygen? Where do we want to drop it off? We want to drop it off to our cells, to our tissues. They're the ones that need the oxygen. So look at this. We have two places, lungs and tissues. Do you think they're the same temperature? And before you answer, think about this. Lungs are full of air. And the air is coming from outside, and it's usually cooler than your body temperature. You'd be really hot if the temperature was the same or higher than your body temperature. Tissues, on the other hand, are metabolizing. In other words, they're working. And work makes you produce heat. So tissues tend to be hot. So look at this. Without changing anything, if I go to the lungs, are they warmer or cooler? They're cooler. If temperature goes down, the curve shifts to the left. Well, we already know if the curve shifts to the left, what does that make it easier to do? Does it make it easier to load or easier to unload? It makes it easier to load. So look, we didn't do anything at all, but because the lungs are cooler, we can pick up more oxygen. And then now we're going to go to the tissues. So the tissues, remember, they're the ones that are doing the metabolism. They're warmer. What happens to the curve if you get in a warm temperature? Which way does the curve shift? It shifts to the right. If we increase the temperature, the curve shifts to the right. And if the curve shifts to the right, what does that make it easier to do? Load or unload? It makes it easier to unload. And so look, we didn't do anything at all. Just because the cells, the tissues are warmer, it's going to be easier to drop off oxygen. But not only that, look at this. Think about different tissues. So I'm going to go to the gym. And when I go to the gym, I've decided today I'm only going work out my arms. That's it. That's all I'm going to do. So here's this person, and he's going to work out his arms right here. So the legs, he's just going to stand. They're not going to be doing anything except holding him up. But he's got dumbbells in these arms, and he's working them out. So think about this. Do you think his arms and the rest of his body are going to be the same temperature? When you work out the arms, what's going to happen to the temperature in the arms? It's going to go up. They're going to get hot. So look, all in here, we're going to increase the temperature, not down here, just here. So when the blood goes to your arms are hotter than they usually are. In other words, we increase the temperature. And if we increase the temperature, which way does the curve shift? It shifts to the right. And what happens if you shift the curve to the right? What happens to affinity? Affinity goes which way? Affinity goes down. And if affinity goes down, what does it make it easier to do? Makes it easier to unload. So look, these muscles, they're the ones doing the work. They're the ones that need more oxygen because they need to make ATP. But just by the fact that the temperature has changed and the temperature is warmer, I'm going to drop off more oxygen to those tissues, those cells. What about down here? They're not as warm, and so they're not working as hard. They don't need as much oxygen, so I'm going to drop off less oxygen down here. Does it make sense to everyone? Any questions about it? So look at that. Look, increasing the temperature causes the curve to shift to the right. Shifting the curve to the right decreases the affinity for hemoglobin has for oxygen. And so that's going to make it easier to unload oxygen. So any tissue that's hot, any tissue that has heat, I'm going to deliver more oxygen. But look, what happens when I get back to the lungs? The lungs are cold temperature again, or lower temperature. What's going to happen to the affinity? Curve's going to shift to the left. What's going to happen to the affinity in the lungs? Affinity's going to go up. If affinity goes up, what's it going to be easier to do? It's going to be easier to load. So because the lungs are cooler, I can load more oxygen. And because the tissues are warmer, I can unload more oxygen. Does it make sense? And so it looks like this. So lungs are over here. Tissues are over here. Not only that, but it's dose dependent. What that means is that the warmer it gets, the more oxygen I drop off. So look, here are 3 temperatures. And look, at this temperature, I'm not going to drop off much at all. But at this temperature, I'm going to drop off more. And this temperature, I'm going to drop off even more. It's dose dependent. So what happens if you get a fever? What happens if you get a fever? What happens to your body temperature? goes up. All over your body, it gets hot. So if you have a fever, what's going to happen to the curve? Which way is it going to shift? It's going to shift to the right. And if it shifts to the right, what are you going to do? Drop off more oxygen or drop off less? You're going to drop off more. Well, if you have a fever, it probably means you're sick. And your tissues are going to need energy in order to fight off whatever it is. And to make energy, you need more oxygen. So that fever helps you fight it off by delivering more oxygen. Make sense? So if we change it or increase the temperature, it shifts to the right. If we decrease the temperature, it's going to shift to the left. So lungs are cooler than tissues, and so it's going to be easier to load. Again, it's dose dependent. So the more you move this way, the more you can load. Any questions? Is it making sense? So that's one thing that makes the curve shift. Let's look at the others. So when I talked about other things that can affect it, there was carbon dioxide, and then there was pH, and then there was hydrogen ions. Well, these are very, very closely connected. In fact, they're so closely connected, you can't separate them. And this is why. If you put carbon dioxide in water, just like putting it in blood, some of that carbon dioxide will chemically combine, and it'll form something called carbonic acid. So look, we had a carbon, we had a carbon. We had 1, 2, 3 oxygens, we had 1, 2, 3 oxygens. We have 2 hydrogens, 2 hydrogens. Carbonic acid. It's an acid. And like all acids, acids give up or release hydrogen ions. That's what makes them an acid. So this is going to release a hydrogen ion. And if I take one hydrogen away, I'm going to be left with one. And so what's going to be left is this. It's called bicarbonate. So this happens automatically. If I take carbon dioxide and I add it to water, automatically some of it is going to combine chemically, form this carbonic acid, and that carbonic acid is going to dissociate, release a hydrogen, and what I'm going to be left with is a bicarbonate ion. Well, look, a hydrogen ion looks like this. And a hydrogen ion is what makes an acid an acid. So the more hydrogen ions you have, the stronger the acid. Well, we want to be able to measure how strong an acid is. So what we do is we measure the hydrogen ions. And when we measure the hydrogen ions, that's what gives us pH. So pH is just a scale. It goes from 0 to 14. Right here's 7, which is neutral. It goes from 7 to 14, and all it is the concentration of hydrogen ions. But it's a logarithm, and it's actually 1 over the logarithm. which makes it negative. So if I take the hydrogen ions, which I got from here, I take the logarithm, but you don't have to worry about that. But look, it's in the denominator here. If this number gets bigger, the pH is going to get smaller because it's down in the bottom. So if I take carbon dioxide, I add it to water, I'm going to get hydrogens. So in other words, the more carbon dioxide I have, the more hydrogens I have. And remember, the scale for pH is 1 over the hydrogens. So if this number goes up, pH is going to go down. They're very tightly connected. You can't separate them. So automatically, if I increase carbon dioxide, I will increase hydrogen ions, and I will decrease pH automatically. So in other words, or the opposite of that's also true. If I decrease carbon dioxides, so look, if this number goes down, that number's got to go down because that's what I'm making it from. So automatically, I'm going to decrease hydrogen ions, and I'm going to increase pH because remember, it's in the bottom of the equation. Here's that. Automatically, if I increase carbon dioxide, I'm going to increase hydrogen ions, and I'm going to decrease pH. Automatically, if I decrease carbon dioxide, I will decrease hydrogen ions, and I will increase pH. Does everybody understand where I got this? Anybody have a problem with it? Again, it's because of this equation. So remember, carbon dioxide combines with water, and we get carbonic acid. And carbonic acid dissociates into a hydrogen ion and bicarbonate. I'm getting writer's cramp. So look, if this number goes up, this equation is going to go this way. That also has to go up. So automatically, if I increase carbon dioxide, automatically, hydrogen ions are going to go up. And just because pH equals 1 over the hydrogen ions, pH is going to go down. So we get that. And then just the opposite too. This is something you should just memorize. It's something that you should just absolutely know. And you should know it like being able to know your face, recognize it in the mirror. Just as well as you know your face, you should know this. Because if you stay in healthcare, you will use this the rest of your life. It's not something you're going to use and just you should forget it. You're going to learn it and forget it. You're going to use it over and over and over and over and over because it has to do with respiration, but it also has to do with the kidney. It has to do with metabolism. It has to do with all kinds of things. It's a really, really important thing for you to learn. And you shouldn't have to sit down and figure it out like we did. You should be able to do that, but you should also just know that if I increase carbon dioxide, automatically I'm going to increase hydrogen ions. And because of that equation, if I increase hydrogen ions, I'm going to decrease pH. And you should know that if you decrease carbon dioxide, you are going to decrease hydrogen ions. And because of that equation, you're going to increase pH. It's something you should just memorize. So when we talk about it, we can talk about it together because they're so connected. You cannot separate them. They're so tightly connected to each other. So we can talk about them all at once. So if I increase carbon dioxide or I increase hydrogen ions or I decrease pH, that's any one of these is going to make the curve shift to the right. The curve shifts to the right. Well, we already know if the curve shifts to the right, what does that do to affinity? Decreases the affinity. And if I decrease the affinity, what does it make it easier to do? Makes it easier to unload. Exactly. So look, any one of these will do that. So let's look at this. By the same token, if I do it in the other direction, it's going to shift to the left. So if I decrease carbon dioxide, or I decrease hydrogen ions, or I increase pH, it's going to shift to the left. And if it shifts to the left, it's going to be easier to load. Well, let's don't forget, where are we loading and where are we unloading? Remember, we load oxygen in the lungs, and we unload oxygen in the tissues. Well, we already know that we have different amounts of carbon dioxide. Remember, in the lungs, the partial pressure of carbon dioxide is 40. But in the tissues, the partial pressure of carbon dioxide is 45. So look, when I get to the tissues, there's more carbon dioxide. So what about hydrogen ions? Are they going to be more or less? They're going to be more. What about pH? Is there going to be higher or lower? Remember, they're connected. So look, when I get to the tissues, because there's more carbon dioxide, automatically I have more hydrogen and a lower pH. The curve is going to shift to the right. And if I shift the curve to the right, what does that mean for affinity? Affinity does what? Goes down. And if affinity goes down, what's it going to make it easier to do? Load or unload? So look, here's the tissues right here. The tissues are high in carbon dioxide. And then when I get back to the lung, think about it. This is partial pressure carbon dioxide of 45. But when I get back to the lung, the partial pressure of carbon dioxide is 40. So in other words, carbon dioxide went down. So what happened to hydrogen ions? They went down. What happened to pH? It goes up. So remember, they're tightly connected to each other. And we know that if we do any of this, the curve is going to shift to the left. And if it shifts to the left, what happens to affinity? Affinity goes up. And if affinity goes up, what does that do to the ability or to the What does it make it do? Easier to load or unload? Load. So look, here's the lungs right here. So just because there's less carbon dioxide in the lungs, it's easier to load. And because there's more carbon dioxide in the tissues, it's easier to unload. Does it make sense? But remember, we had a guy, and don't forget, carbon dioxide and pH and hydrogen ions are all connected to each other. So if I look at this graph, it looks exactly like this graph. They're the exact same graph because you can't separate those. In fact, usually we just put them all on the same graph. So look, carbon dioxide, hydrogen ion, pH, all right there. Same thing here, same thing here. Because you cannot separate those things. But remember, we had this guy at the gym. I guess it was me, but anyway, the gym. And here he is. Remember, he was doing biceps curls. And so not only are his arms going to get hotter, but also when those muscles in the arms work, they're going to produce more carbon dioxide. So there's going to be more carbon dioxide here and down in the legs. More carbon dioxide, more hydrogen ions, a lower pH. And so this curve is going to look like this one over here. And so the curve is going to shift to the right. So in the arms, nowhere else, but in the arms, the curve is going to shift to the right. And I'm going to be able to unload more oxygen. But if I look down here in the legs, they're not working. There's less carbon dioxide. There's less hydrogen ions. And there's a higher pH. And so the curve looks like this. And so I don't unload as much because those muscles don't need it. Same thing's true for tissues and lungs. So here's the lungs. There's less carbon dioxide. Here's the tissues. There's more carbon dioxide. Any questions? It's also dose dependent. So if you have a even further this way or further this way, so if you work your muscles even harder, this might move over even further. So you might wind up with a curve over here, which would mean you drop off even more oxygen. Okay, there's one more thing that affects curve shifting, and that's this stuff called BPG. Now, unfortunately, BPG is also called DPG. And DPG is also called 2,3-DPG. You don't have to know what the words mean, but I'll tell you. BPG stands for bis-phosphoglycerate. DPG stands for diphosphoglycerate. And then there's 2,3-diphosphoglycerate. What you need to know about it is that it's a byproduct of glycolysis. You remember anaerobic glycolysis? Everybody remember that? Well, if you think about anaerobic glycolysis, anaerobic means no oxygen. So the only time you would do anaerobic is when oxygen was low. What anaerobic is we're going to take a glucose and we're basically going to split it in half. What we're going to get is something called pyruvate. which can be turned into BPG. BPG. So look, the only time we're going to make BPG is when we don't have any oxygen, because that's when we're going to do anaerobic glycolysis. Anaerobic. In other words, there's no or not enough oxygen. Well, if I don't have enough oxygen, and these are the cells right here, they don't have enough oxygen, what should I do when the blood comes here? Do you think I should drop off more oxygen or drop off less? They don't have enough. So should I drop off more or should I drop off less? I should drop off more. Well, if I want to drop off more, I need the curve to shift. So which way should the curve shift? To the right or to the left? It should shift to the right. And that's exactly what it does. If I increase BPG, the curve shifts to the right. If I decrease DPG, that means I have enough oxygen, and the curve shifts to the left. Over here. So if I look at that graph, you can see it. So there's normal. There is shift to the right. I have more BPG. That means because I have less oxygen, I need to unload more. So if your muscles go into anaerobic glycolysis, we're going to shift the curve to the right, and we're going to drop off more oxygen. If it shifts to the left, that means there's a lot less BPG. I don't need as much oxygen. There's no need to drop it off. There's no need to unload. Make sense? Any questions? So that's curve shifting. So if we go back to our first slide, these are the things that cause the curve to shift. Temperature. I didn't go back far enough. So these are the things that cause the curve to shift. Temperature, pH, which is connected to hydrogen ions, which is connected to carbon dioxide. Remember, they're all interconnected. And then the other one is BPG. Any questions about any of those? So what I'd like you to be able to do is interpret one of these graphs. So if it didn't have any labels on it, except you would know, or I would tell you that this one's normal, then you should be able to say that one shifted to the right. So what is this? Well, it could be an increase in temperature. It could be an increase in carbon dioxide. It could be decrease in, or sorry, pH, or an increase in hydrogen ions, or an increase in BPG. Those are the things that are going to make it shift to the right. If this one here didn't have any labels on it, then what would make it move? Well, a decrease in temperature, a decrease in carbon dioxide, a decrease in hydrogen ions, an increase in pH, or a decrease in BPG. Make sense to everyone? Any questions? Now this one has labels on it, but they don't have to. Any questions? Well, I don't think we're going to get through today, which is okay, but that's as far as I think I want to go. So next time we'll pick up here and we'll finish this up. I promise we'll finish it up. We only have about 30 more slides, so it won't be that bad. But make sure that when you study this, that you remember, first of all, what's going to make the curve shift. And these are the things that make the curve shift. And then what happens if you shift left? And what happens if you shift right? And then what causes these shifts? And again, that's temperature, carbon dioxide, hydrogen ions, pH, and then BPG. Any questions? Okay, well, I'll see you next time. So I'm going to go ahead and stop recording, but remember these recordings are available. I also posted some more of them to my YouTube channel. So if you want to look at them from there, they'll also be available there.